Hello everyone, my name is Mahina Akhtar and I am a biology student at Medgar College. My research is TAF1 is the new coactivator of NF-kappa B. Significance and background. How does NF-kappa B regulate myriad of genes and controls cellular response? The transcriptional activation of NF-kappa B is triggered by microbial infections, necrotic cellular agents, genotoxic and oxidative stress, and pro-inflammatory cytokines. Subsequently, the NF-kappa B directed biological functions culminate by inhibiting apoptosis, promoting cell proliferation, and stimulating angiogenesis and metastasis, given that the NF-kappa B controls many target genes. The mechanistic knowledge of molecular linkage between inflammation and cancers remains incomplete. Hypothesis. How does NF-kappa B functions are regulated by transcriptional coactivators? A coactivator is a type of transcriptional co-regulator that binds to an activation to increase the rate of transcription of a gene or set of genes. The activation contains DNA binding domain that binds either to a DNA promoter site or a specific DNA regulatory sequence called an enhancer. TAF1 is a new coactivator of NF-kappa B. We wanted to access if there are new coactivators of NF-kappa B given that it controls myriad of genes. 2 years ago, we performed a screening loose reverse assay. This assay was performed by transfecting NF-kappa B promoter into the 293 T cells. As you can see, comparing 2 and 13 shows a big increase. Domain of transcriptional associated factors. Parkinson's diseases associated with intellectual disabilities, which are X-linked to dysmorphic features, and X-linked dystonia. Kinase phosphorylates the target proteins on amino acid serines, threonine, and tyrosine. HAT, histone acetyltransferase. It accelerates the target proteins on amino acid lysine, bromodomain. It binds to acylated lysine on the target protein. TAF1 anti-cane domain activates NF-kappa B. TAF1 is a multi-domain protein, so we wanted to test which domain of TAF1 activates NF-kappa B motor using luciferase assay. We generated the constructs for each domain and then transfected each domain with NF-kappa B promoter and the individual constructs. The result shows that N-terminate domain of TAF1 activates NF-kappa B. Kinase is a protein that functions as an enzyme to phosphorylate the target protein. TAF1 phosphorylates serine 28 on histone H3. To understand what targets are phosphorylated by TAF1, we treated the cells with TNF-alpha, LPS, IL-1 beta, IL-1 alpha, IL-6, and IL-8. Microarray and heat map of genes regulated by TAF1. To understand the significance of TAF1 in NF-kappa B, we knocked down the THP1 cells and treated the normal cells and TAF1 knockdown cells with TNF and LPS. As you see on the right, first THP1 cells, second knockdown TAF1 with RNA, 3 treated with TNF and LPS, 4 RNA extraction, 5 microarray, 6 heat map analysis, 7 compare with normal cells. Number of genes affected by knocking out TAF1 and THP1 cells. TAF wild-type TNF, 54 positive regulated, 29 negative. TAF knockout TNF, 36 positive, 46 negative. TAF1 wild-type LPS, 55 positive, 29 negative. TAF1 knockout LPS, 53 positive and 30 negative. TAF1 affects all the NF-kappa B target genes across the chromosome circuit plot. So this shows that the genes are distributed across the chromosomes from 1 to 22. TAF1 knockdown affects NF-kappa B pathways. As you can see, this analyzes genes that are part of NF-kappa B using KEGG pathway. TAF1 works with transcriptional machinery to activate NF-kappa B. We propose a model, TAF1 interacts with NF-kappa B, and it shows how TAF1 regulates myriad of NF-kappa B genes. I thank the CRS program, Dean Patwari, Dr. Mukesh Gautam, and my mentor, Syed Mujaba. Thank you so much.